secondary structure is the local spatial structure of the main chain in a selected segment of a polypeptide chain. Depending on the numerous non-covalent interactions, the whole polypeptide chain falls into a tertiary structure. The tertiary structure refers to the overall three-dimensional arrangement of all atoms in a polypeptide chain. In the complete folded tertiary structure of a protein, amino acids that are far apart in the polypeptide sequence and in different types of secondary structure may interact by several kinds of weak interactions and sometimes by covalent bonds such as disulfide cross-links. For example, myoglobin is the first protein whose three-dimensional structure was elucidated. Myoglobin is a relatively small oxygen-binding protein in muscle cells. It functions both to store oxygen and to facilitate oxygen diffusion in rapidly contracting muscle tissue. Myoglobin contains a single polypeptide chain of 153 amino acid residues and a heme group. This figure shows the structure of the myoglobin. The backbone of the myoglobin molecule consists of 8 relatively strict segments of alpha helix from A through H, which are connected by bands, some of which are beta terms. More than 17% of the residues in myoglobin are in these alpha helical regions. The red group surrounded by protein is heme. The capacity of myoglobin to bind oxygen depends on the presence of heme. The plate heme group rests in a crevice or pocket in the myoglobin molecule. This picture is the heme group present in myoglobin, hemoglobin, cytochromes, and many other proteins. Heme consists of a complex organic ring structure called protoporphyrin, which binds an iron atom in its ferrous state. The iron atom has 6 coordination bands, 4 in the plane of and bonded to the plain porphyrin molecule, and 2 perpendicular to it. In myoglobin and hemoglobin, one of these is bound to the R group of the histidine residue at position 93, also designated his F8. The other is open and serves as the binding site for an oxygen molecule. Within the pocket, the accessibility of the heme group to solvent is highly restricted. This is important for function because free heme groups in an oxygenated solution are rapidly organized from the ferrous form, which is active in the reversible binding of oxygen, to the ferric form, which does not bind oxygen. Studies show that the tertiary structure of myoglobin is largely stabilized by the hydrophobic effect between amino acid side chains. Most of the hydrophobic R groups are in the interior of the molecule, hidden from the exposure to water. All but 2 of the polar R groups are located on the outer surface of the molecule, and all are hydrated. The only polar residues inside are 2 histidine residues, which play critical roles in binding iron and oxygen. The three-dimensional structures of thousands of proteins have now been reviewed, which give us the chance to understand the correlation between protein structure and function. In a complete three-dimensional structure, the polypeptide chain can fold to form some special structural patterns or elements. Next, let's discuss the 2 important structure patterns, the motifs and the domains. The motif is also called a fold or supersecondary structure. A motif is a recognizable folding pattern involving 2 or more elements of secondary structure and the connection between them. This structure element can fold against each other in space. A motif can be very simple, such as beta-alpha-beta loop or alpha-beta term alpha, and usually represent only a small part of a protein. A motif can also be a very elaborate structure involving scores of protein segments folded together, such as the beta barrel. In some cases, a single large motif may comprise the entire protein. Note that a motif is not a hierarchical structure element falling between secondary and tertiary structure. It is simply a folding pattern. The synonymous term super-secondary structure is thus somewhat misleading because it suggests a hierarchy. The motifs can exert special functions in proteins. For example, calcium-bonding proteins usually contain the calcium-bonding motif, which is an alpha-loop alpha motif. There are several conservative polar side chains in the loop, which can bind with calcium ions by hydrogen bonds. Another example is the zinc finger motif. This structure composed of an alpha helix and two antiparallel beta strands, which are held together by a single zinc ion, coordinated to 4 residues. It looks like a finger. Several hydrophobic side chains in the core of the structure also lend stability. Many eukaryotic DNA-binding proteins contain zinc fingers, which are responsible for the interaction between proteins and DNA. Some polypeptides with more than a few hundred amino acid residues often fall into 2 or more independent compact structures that may be connected by a flexible segment of polypeptide chain. These compact globular structure units are called domains. Different domains often have distinct functions, such as catalytic activities, the binding of small molecules, or interactions with other proteins. In many cases, a domain from a large protein will retain its native 3-dimensional structure even when separated from the remainder of the polypeptide chain. In a protein with multiple domains, each domain may appear as a distinct globular lobe. More commonly, extensive contact between domains make individual domains hard to discern. Small proteins usually have only one domain. That means the domain is the protein. Okay, let me summarize this section. Tertiary structure is the complete 3-dimensional structure of a polypeptide chain. In a complete 3-dimensional structure, the polypeptide chain can fold to form special structure patterns or elements, such as motifs and domains. A motif is a folding pattern involving 2 or more elements of secondary structure and the connections between them, which can exert special functions in proteins. Domains are regions of a polypeptide chain that can fold stably and independently and exert distinct functions. Okay, that's all. Thanks for joining. See you next time.